Hello class, this is Dr. Branch. I'm about to discuss Christian ethics and genetic engineering. I'm going to give a very quick overview of an infinitely complex subject. My slide deck is far too large for an 18-minute time slot, but I do want to introduce you to a few central and key ideas related to genetic engineering. And to do that, I'm going to begin by contrasting two stories, one which is true and one which is fictional. So the fictional story involves a young girl named Ashanti de Silva, who in 1990, Ashanti became the first person to receive genetic therapy. She suffered from a very debilitating disease which affected her ability to respond to different diseases. Her immune system was compromised. And so scientists were able to replace these compromised T cells with T cells which had been genetically modified and dramatically improved her ability to fight diseases. Now, she still had to continue getting medication throughout the rest of her life, but she most likely would have died. This was a profound change. The very cells within her body were changed, and I have a detail of how that occurred in your class notes. And she's a thriving person today. In fact, earned a master's degree from Ohio State and is now a genetic counselor. So this is the idea of genetic therapy, 1990, 30 years ago now, but the very promise and the hopeful and the good side of genetic therapy, which we as Christians would affirm. On the worst extreme is the fictional story Jurassic Park by Michael Crichton. And you remember the stories of DNA from mosquitoes trapped in amber is modified to try to and inserted into frog eggs to try to give birth to new dinosaurs. But when things get out of hand, the entire island is overrun by dinosaurs. And I realize it's science fiction, but it does show in a very extreme view, this fictional story, the dangers of tampering with the unknown. What happens when we begin to play around with human DNA? So with that said, I have quite a bit of information in my notes about what DNA is. And again, much of this is for my own reflection as well, to help keep me oriented and to keep me informed. I won't go through all of it, but just to say a few things, it is infinitely complex but relatively simple all at the same time. Some of you guys, if you've ever written code for computers, it's basically binary code. DNA is very much like that. A's and T's and G's and C's lined up, these four base pairs. And the A's always lined up with T's and the G's always lined up with the C's, these fascinating bases. Arranged in a double helix or a winding staircase. It's infinitely simple yet fantastically complex all at the same time. This is actually a picture of what a strand of DNA might look like taken with an electron microscope. Isn't that amazing? So the structure of DNA, I won't go through this. I'm assuming most of you learned this in junior high or high school biology, but merely to point out that this is the stuff of which we're dealing with, and this is how God has used, these are the tools that God has used to design us as humans and to pass on our life to other humans. So I'm going to skip through a great deal of this. You can read it on your own. But I do want to point out just one quick thing to you. When we talk about genes, genes are a distinct stretch of DNA that codes for something special about who you are. Originally, people thought that the human genome would include around 100,000 genes, and now we know it's much smaller, actually. It's about 30 to 40,000 genes. I hear the number 30,000 more frequently. So I'm not a geneticist. My point being, the vast majority of your DNA is actually what we call intergenic DNA, and it doesn't code for any specific traits. Only about 2% of the DNA within inside of us codes for specific traits. So it's amazing how much work God gets out of just a little bit of DNA. So let's talk about recombinant DNA. This is when DNA is modified in the laboratory for specific purposes, either for research or by scientists to produce some therapeutic cure. One of the most well-known examples is the creation of these bacteria that produce insulin. Insulin used to be very, very expensive, but a number of years ago, recombinant DNA was used to transfer into this bacteria, which is now used to produce insulin in the lab, and it's helped lower the cost of insulin and make insulin much more available for diabetic folks. If you have a friend who has, or perhaps one of you has the diabetic pump, then you are a beneficiary of this sort of technology. Again, something like this is very fascinating, and we as Christians in our drive to have mercy and compassion for suffering people and to be the hands and feet of Jesus. We affirm these sort of things, but it does pose some other problems when we start moving beyond some of this. So epigenetics, I want to say a quick word. Your DNA within your body cannot be changed. So when you're thinking about the human genome, the DNA within you, I say it can't be changed. It can't be changed by your behavior. That's what I'm trying to say. Your behavior, the things you do in life, doesn't change your DNA. However, the epigenetics is a fascinating new field of science. I won't spend a lot of time on it, but it is somewhat riveting. The epigenetics are what actually tells your DNA to turn on and turn off. It's like knobs and switches. It turns things in your DNA off and on and up and down. And what you need to know is the DNA can't be naturally modified by our behavior, but the epigenetics, in fact, do or are affected by our behavior. There's some fascinating research about drug and alcohol abuse related to epigenetics, which is informative on a lot of other human behavior and how epigenetics works. The epigenome are basically chemical tags on the back of your DNA that tells certain genes to code and certain genes to be quiet. So if you've ever wondered, if you think about it for just a second, it's very fascinating. In every cell of your body, you've heard this, there's all the DNA to replicate you, but have you ever thought about why a skin cell only does skin cell work, why a blood cell only does blood cell work, why a brain cell only does brain cell work? These are really fascinating questions. The DNA to make an entire another me or you is there, but those cells only turn on a little part of that DNA. It's all related to the epigenome. The epigenome tells that skin cell, just do skin cell stuff, tells that blood cell, just do blood cell stuff, and all the intricate. I'm oversimplifying because I'm not a geneticist. I'm a professor speaking to students at a seminary, but what I want you to remember out of all of this are these 2 points right here. The DNA can't be naturally modified by our behavior, but the epigenetics, which turns on and off and up and down these genes within our body, that can be changed by human behavior. That's what I want you to remember out of this. So genetics, disease, and therapy. Let's talk about I have a lot of information about how diseases are passed on genetically, dominant traits, recessive traits. I'm assuming for graduate students that your biology class in college taught you these things. I only mention them here to emphasize some of the most frightening diseases we deal with in life are genetic diseases. Things like Huntington's chorea and Huntington's disease. Recessive diseases like Tay-Sachs, sickle cell anemia, which can be so devastating. Sex-linked inherited diseases are only passed on by the X or Y chromosome. They can be quite befuddling as well. Muscular dystrophy, hemophilia. So these are things that strike terror in the heart of most of us when we think about disease, and we don't want to suffer them. We think about our children getting this disease. We don't want our children to suffer from these diseases. When we have a child suffering, most of us would cry out to the Lord, Lord, give it to me. I don't want my child to go through this. I would rather go through it than my child. So what we are talking about is an effort to eliminate some of these genetically inherited diseases. But what I would remind you is that there are many diseases which are genetically influenced, but they're not genetically caused. There's a very difference there. So there are the diseases with a genetic component is what we're getting at, and it's difficult to understand how those genetic components work, but some people may have a genetic predisposition that sets them at a higher risk for certain forms of cancer or heart disease or things like that. It doesn't mean they are going to get cancer or they're going to get the heart disease, but what it does mean is if other environmental factors are weighed in, then this person might be at a higher risk for this because of a genetic predisposition. There's some of this going on with human behavior as well, but I won't get into that right now. That said, what if we could eliminate all these? Wouldn't we want to do this? So this is what's actually going on, and so what's being done now is what's called genetic testing. This can be done on humans as adults, but it's also can be done on embryos. And so some people with a disease that is genetically passed down, such as Huntington's, they will actually have embryos tested to see if the embryo has the gene for Huntington's disease, and those embryos in some cases will be discarded. And so couples will have in vitro fertilization, what we talked about last week, multiple embryos created in a petri dish, and then they'll choose one that doesn't have the Huntington's gene, and the rest will be discarded. They'll die. So this poses an interesting question. It's a moral question. Should these human life be created knowing that if they have certain traits and certain diseases, then we are going to eliminate them? It's a very painful discussion. Genetic counseling is basically what happens. It's people who help folks process the genetic information they've received about themselves or their children. Genetic enhancement is fascinating. This is tied very, very closely to what we're about to discuss, this CRISPR technology, but it refers to changing someone's genome not for therapeutic reasons, but for enhancement reasons, to make someone stronger, faster, more beautiful. So it means just what it says, enhancement. It's not that there's something wrong, but the person would like to have more muscle mass. The person would like to have greater memory. Perhaps a distance runner would like to have more particular ability to process oxygen and run long distances. I would love to have more fast twitch fiber, be able to be fast. Well, that's what genetic enhancement is all about. And so the CRISPR technology has made this feasible to have serious discussions about whether or not we can enhance humans, not just cure disease, but somehow make humans better, assuming that we as humans know what a better human looks like, right? That's part of our sinful nature. Gene therapy is exactly what happened to little Ashante da Silva, who's now a grown woman. So it's changing genes in order to prevent or heal a disease. There's two types of gene therapy. I want you to keep this in mind. So this is probably going to be on your final. There's a lot in this slide deck, but you do need to know this. Somatic cell gene therapy is what happened to Ashante. She's already born, and so cells in her body are changed. She doesn't pass on those changes to future generations. It simply helps her fight infections better today. Germline gene therapy is when you actually alter the sperm or their egg cells so that someone passes on what are considered to be more favorable traits to a next generation. That might be eliminating the possibility of passing on a disease. That might be enhancing your future generations so they could all be fast or strong or smart. So I have a slide here that shows the difference between those two. When I post the slide deck, you can take a look at it. I think it will help you understand it. Oh, even nanotechnology. There's serious discussions of using these little nano robots to alter, do this work of genetic research. It is absolutely fascinating. I'm going to skip past the Human Genome Project because it's done. You've seen a video for the class that I posted this week about CRISPR, so we're going to move it past that. Xenotransplantation is when you take human DNA and put it into animal DNA with the hope that those animal organs can then be used to transplant into humans. So you might have a pig that's developed to have a heart that someday could be transplanted into humans, have just enough human DNA that the human body wouldn't reject it. So this is also tied into cloning. If you had an animal that was able to do such things, then very much so you'd want to clone that animal because you can make a lot of money from it. I'm going to skip past the legal cases. Let's move to a conclusion. I'll say a few words about possible benefits and dangers, and we'll talk about these things in more detail in our class discussion. But I have this quote from Mary Shelley in her opening introduction to Frankenstein. And she said, when she described writing the story, she said, I saw the hideous phantasm of a man stretched out, and then on the working of some powerful engine, show signs of life and stir with an uneasy half-vital motion. Frightful it must be, for supremely frightful would be the effect of any human endeavor to mock the stupendous mechanism of the creator of the world. So Mary Shelley actually said that the book Frankenstein was an attempt to say, well, this is what can go wrong when humans try to step into the place of God. Now, I'm going to try to define that phrase, playing God, in just a moment. We have to be careful. It can be overused. But I think she raises a point here which needs to be considered, and I believe it's relevant when we consider genetic research and genetics. So, gene isolation. Francis Collins, who is, in fact, a born-again Christian, says this. He was the head of the Human Genome Project for years. He said, "Gene isolation provides the best hope for understanding human disease at its most fundamental level. Knowledge about genetic control of cellular functions will underpin future strategies to prevent or treat disease phenotypes." So this is a very smart man saying what he thinks the most beneficial thing that's going to come out of all this research is to be able to isolate particular genes where we say, okay, this gene contributes to that disease, this gene causes that disease, this gene is a factor in this disease, and then to be able to target those genes. So one of the challenges is you can also, let me go ahead and scoot on and talk about some of the dangers. We'll skip past Brave New World. One of the dangers is denial of coverage. You are aware that medical costs have gone through the roof. They're skyrocketing. So there's a fear that if insurance companies could get their hands on your your DNA template and discover that you are predisposed for something like Huntington's, they might say, we'll cover everything, but we won't cover that. So there's this fear of denial of coverage, which is not legal in the United States, but of course, it's a fear that something like that could happen. Stress and depression, would you want to know that you're going to get Huntington's? Environmental concerns. What if certain plants or animals are so modified that they are no longer resistant, specifically plants that are resistant to things like Roundup, which kill weeds? What if you have a weed that was no longer resistant to poison? That could be a real challenge. So let me give you some religious and moral reflection. The sanctity of human life should be our primary concern, because if one affirms the sanctity of human life, then genetic engineering that destroys innocent human life is immoral. With that in mind, I'm going to say this as well. Be careful about saying, well, anything we do is playing God, because I believe God's sovereign. He's completely in control. And to that degree, because he's completely in control, we're never going to affect his sovereignty. He's always going to be in control. But so often that whenever there's a new medical intervention developed, people say, aha, that's playing God. But think carefully. Are we playing God when we take aspirin before a meal, or if we have a headache? Are we playing God if we take medication to control high blood pressure? Most of us would say no. So what does playing God mean? Playing God involves humans taking a likeness to God for themselves, which God did not intend. So we are extending our autonomy into areas which God did not intend. I don't think any of us would say taking an aspirin is playing God, is my point. But then there are other areas where God did not intend us to do. Scott Ray says that playing God refers to human beings usurping prerogatives that belong to God alone, such as the determination of and the timing and manner of our deaths. That's one example. So let me give you some suggestions for guidelines. This is from my friend Barrett Duke, who is now working with Southern Baptist in Montana. He's a very, very bright man. But he suggested these guidelines for genetic engineering. And I want to stress the last one. If we're going to do any of these processes, that there's verified zero chance of the introduction of new mutations that can be expressed in future generations from that germline. And what he means is, if we're going to pass along these traits to the next generation, and we hope they're favorable, we have to make sure that whatever it is we're passing along doesn't have unintended negative consequences that produces a whole new set of problems. And this is bracketed by his emphasis, which I share, on the sanctity of human life, that we're not going to create embryos just to destroy them. This is some thinking on the sanctity of human life and genetic engineering. I hope it's been helpful for you.